So today we're going to start talking more specifically about our microbial organisms. So up until this point, we've been talking about things that are pretty general, cell material, growth requirements that are similar to prokaryotes and eukaryotes. But in this chapter, we're going to focus on just our bacteria. So we're going to start talking about some of the prokaryotes and some of the diseases that you've probably heard about in your other classes or just in your daily living. So remember, prokaryotes are divided into two main domains. So we have our bacteria and we have our archaea. So two separate domains. What's the difference between the two? Anybody remember? Excellent. The composition of the cell walls. Excellent. So the composition of the cell walls is going to be the difference. And so with that in mind, with that in mind, then, remember that's going to be the variations that we see between those two organisms. Other than that, they're going to be the same. When we look at them, they're similar. In this chapter, there's so many prokaryotes that we can talk about. It's hard to narrow down which ones to discuss. So what I've been focusing primarily on are the pathogens. Now, remember, are there any pathogenic archaea out there? No. Remember, the archaea, when they evolved to fit more with the environment. They evolved to not depend on other life forms. And so because of that, they tend to be more self-living, and so they tend to not cause disease because they don't need us for anything. The bacteria, on the other hand, some evolved to live just in the environment, and some evolved to, unfortunately, be pathogens, and those are the ones that we'll be focusing on primarily in this chapter. So as we start to go forward, what we can see is our prokaryotes then, they can be divided into further groupings. So we can say, okay, we can be either an archaea or a bacterium. And again, the differences between those are the cell walls, whether they have peptidoglycan or that pseudomarine layer. Once we get into the bacteria, we can divide again into gram-negatives or gram-positives. And then from there, we can divide our gram-negatives into the proteobacteria group and the non-proteobacteria group. We'll talk about what that means today. And then our gram-positives can be divided into low GCs and high GCs. The GCs indicate DNA bases. So what we found is that some of the gram-positives, naturally in their DNA, will have a higher GC binding content. So when we sequence them, we just see it like predominant, 80% are Gs and Cs. And so those tend to be in the high GC group. And then the other ones are going to be in our low GC, which means they would be predominantly As and Ts. So let's start with our gram-negatives, and let's start with the proteobacteria. So the proteobacteria, this is really our largest group of bacteria that are out there. There's about 500 different types of genus or genre in this grouping, and there's over 2,000 species. And we're always adding to this, so this number is always changing. Now, to be in the proteobacteria group, you have to be a gram-negative. Remember, we had that division on that previous slide. So if I'm going to be a proteobacteria, I, by definition, have to be a gram-negative. And most of the ones in this group are going to be facultative anaerobes. What does that mean again? They can live without oxygen, but they prefer it, right? If they have oxygen present, they get 38 ATP. And if they don't have oxygen present, they get 2 ATP. So they can grow either way, but they grow faster and prefer to have oxygen. Do you have a question?
They become stronger.
Right, they grow faster. Yeah, there's oxygen present. The reason they're called the proteobacteria group is they were named after the mythical Greek god of Proteus. And Proteus was basically known to be a god that could take many forms. And so when early scientists started studying the gram-negatives, they found that they could have a lot of variations between them. And so when they started talking about grouping them, they decided to name them the Proteus, or the proteobacteria, because of the variations that Proteus was known for. You can see on the side here that the face of the god, it could be a lion, it could be a human, it could be a fox or an eagle. The god could have a mermaid tail, it could have legs. It just depended on its day, whatever day it was happening. So we named them those because of the variation. And in the proteobacteria, we're going to divide them into 5 classes. So we're going to have our alphas and our betas, our gammas, our deltas, and our epsilons. And each of them are going to have unique characteristics. So we get to divide them even further. So let's start with our first group. This is going to be the alphas. And so the alpha proteobacteria are well known to be able to survive in low nutrient environments. So these are going to be ones that are going to be found in areas that are like lakes or streams where nutrients are flushing through quickly and they're not just sitting in an environment-rich area. So they have a couple tricks up their sleeves to be able to help them survive in their environment. One of the things that they have is something called a prostheca. And the prostheca is this little stalk appendage you can see at the end of the cell. There's a red arrow pointing to it. And the prostheca is basically an extension of the cell that allows a lot of our alphas to attach to the surface. It's almost like a suction cup. And so if you think about where they're found, lakes or streams, they're having to deal with currents. Water is washing them back and forth. So having this little extra appendage allows them to kind of anchor themselves. It's almost like a little anchor on their cells. And so this appendage is always present. It's not like one of the pili where they grow it and then get rid of it. If they have it, it's always going to be present on them and they'll attach to it. Here's an example of the Caulobacter then. And Caulobacter is this stalked organism. Again, it's stalked because of that prostheca that is found in a lot of our lakes and streams in this area. For example, if we went to Lopez Lake and took water samples, we would find this little guy. So they're just found in local lakes and regions. One of the big groups that's in alpha is an organism that helps us with carbon cycling. And these are the Pelagibacter. Pelagibacter, when you hear pelagic, think ocean. Pelagic means ocean. So Pelagibacter means that I am an ocean-dwelling bacterium. And these guys are such a big group that they take up about 0.5% of all the prokaryotes in the entire world. Now, I know that doesn't sound like a lot, but remember, we have over 2,000 different species in this grouping. And 0.5% being one section is pretty big when you really think about it. These guys are found all throughout our ocean or aquatic environments. And the way they help us with carbon is they will absorb carbon and take it out of the atmosphere to use it for photosynthesis. You can tell they're naturally green, so that tells us that they're doing photosynthesis. Now, this is important because one of the things we're finding is that we are going through a process of global warming. We can sit here and argue whether it is natural or not, but what we can't argue is that carbon dioxide levels have increased. And the carbon dioxide levels increasing means that they absorb more warmth. CO2 absorbs and holds sunlight in, holds warmth. And that's what's leading to our warmer planet. Now with these guys then, what they do is they pull that CO2 out of the atmosphere. They act as a sink or a reservoir. And then they use that CO2 later for their photosynthesis. So if we didn't have them, we would actually have our planet getting hotter faster. So having them here is actually slowing down some of the global warming that we're seeing. Alphas can also be very important when it comes into agriculture, when we're trying to grow plants. Some of the alphas that are out there are also able to do nitrogen fixation. So we mentioned this before. Nitrogen fixation is where they remove nitrogen gas, N2, from the atmosphere, and they convert it into a usable form. So it can be converted into a nitrate or a nitrite. That would be nitrogen fixation. And those are the forms that we can use and other organisms can use. So it turns out that some of them, for example, rhizobium, is an organism that can help plants grow, because what it'll do is it'll attach to the roots of the plants. You can see the plants have kind of swollen areas on them. We call those root nodules. Those root nodules will swell because inside of them are all these bacteria. And what they'll do is they absorb nitrogen from the atmosphere, convert it into a usable form that the plant can use to make proteins now, and in return, the plant will give the little bacteria sugar. So it's kind of like this win-win situation. We used to think that these were found kind of just randomly in soil, but what we're learning is that plants actually call the bacteria to them when they're first establishing their roots. So as their roots are starting to spread out, they release chemicals and they release sugar particles that draw the bacteria towards them. And once the bacteria come in, their roots curl around the bacteria and then slowly do an overgrowth over them to trap them, to create that root nodule. So it's like the plants have this whole plan. They know that they need these guys. We found that when plants have these root nodules, their growth can be as high as 50% faster than plants without them. So it's really good for them to have them. It's a lot less work for them to spread. Now, not all alphas are nice though. There's another organism out there called Agrobacterium. And this is one that is unfortunately inserted into a plant cell through a plasmid. So remember, plasmid is just a piece of naked DNA. So it's a piece of naked DNA that's out there. And when this agrobacterium absorbs this plasmid, it makes it pathogenic. And so what it'll do is it will start to harm the plant. As it grows in the roots, it starts to create tumors. It starts to cause the plant cells to become infected, and they start going through massive mitosis. And so it actually causes the plants to create kind of this tumorous, or what we call a crown gall, at the base of their roots. So if you see them, you'll see them kind of like right at where the roots are starting to take over in the soils, you'll see kind of this tumorous appearance. And that's usually caused by agrobacterium. So if we look at our alphas then, we can start to separate our alphas. We can separate them based on where they're found and what kind of lifestyle they have. So the first question, of course, that we always ask is, can they cause disease in humans? So if we start to look at our alphas, we have a side that says no, that would be the minus, and we have a side that says yes, and those would be the pluses. So on our plus side, we have a couple of ones that are unfortunately carried by insects. These are going to be ones that are going to be carried, for example, like Rocky Mountain spotted fever, or ones that are going to be like brucella that can be carried through the transmission of an insect bite as well. We'll talk about that in a minute. On this side, though, we have our alphas that are not carried to cause human disease, but instead can cause plant disease. We have plant pathogens, or we have ones that just live in the environment. Now, I'm showing you these tables to kind of give you an idea of where the organisms are found. On your exam, though, I would not have a table and have you fill it in. This is just to kind of give you an overview so that when you're studying, you kind of understand where we're going with them, okay? So don't panic. You don't have to memorize these and fill these in on the exam. They're just to give you an overview. So let's talk about the alphas that are dangerous to us. So the alphas that are dangerous to us are ones that, for whatever reason, have evolved to not be able to make their own ATP. They are unable to do chemiosmosis, so they don't have an electron transport chain. And they're also unable to do substrate level phosphorylation, where they just have an ADP and a phosphate come together. They're not able to do either of those. So instead, they are obligate parasites, meaning that they have to be inside of a host to have that host make ATP for them. So as you can imagine, they don't survive very long on their own. Rickettsia is an example of this. It can only reproduce inside a mammal's cells. If I try to grow this organism on a petri dish, it's not going to work. It has to be inside mammalian cells. These are generally transmitted through bites of insects, primarily ticks. So this would be, for example, one of the fevers that we hear sometimes about in the news, which is Rocky Mountain spotted fever. It's called that because it was found predominantly in the Rocky Mountain region. Again, we're trying to move away from naming diseases after the locations where they're found, because we don't want to always try to make an area sound bad, number one, but also these diseases can spread. And so then they're no longer just in Rocky Mountain. So Rocky Mountain spotted fever is unfortunately something that can cause these petechial rashes. And you can see a picture of a petechial rash. If you look at the surface of the skin, if you were to touch it, it feels smooth. It doesn't feel like there's anything there. But if you looked, you'd see like these little like sunburst kind of appearances on them. And that's what we see here. And that sunburst would be kind of a red in color. What this is this is where this organism is bursting open blood vessels and bursting open red blood cells and stealing the ATP that's inside of them. So it causes this kind of blotched appearance on our skin. So when you see a petechial rash, usually the patient, it doesn't hurt, it doesn't itch, it's actually under the surface of the skin. But what we're seeing are the effects of lysing of blood cells and blood vessels, which again can be very harmful. The reason they're doing that, they're trying to steal as much ATP as they can. Now, rickettsia can be treated. If you get to a hospital, they can put you on antibiotics that will clear the infection. However, we are finding that sometimes people will have long-term issues, kind of like with Lyme disease, where they can have issues where maybe there was tissue damage from this kind of hemorrhaging. Maybe there was damage to the cells that take time to recover. The other concern is in areas where Rocky Mountain spotted fever is high, like in Utah, Colorado, areas like that, we run into issues where a lot of times people just get reinfected right away. And so we're still trying to figure out how to get a better handle on our tick population to try to get this guy under control. Another alpha that can pose problems to us is one that's referred to as cat scratch fever. You might have heard that one. It's an organism that's genus is Bartonella. And you will want to learn the organisms that we're talking about. So like when I give you examples here, you will want to learn these. Bartonella is the causative agent of cat scratch fever. It's usually spread through a cat bite or a deep cat wound. So these are usually defensive wounds. This isn't like when you're playing with your cat and they just lightly scratch you. This is usually where like the cat was scared or was being handled roughly and was biting or trying to get away. Where we see them a lot is usually in children. And children who were either handling cats or kittens too roughly. So they were like squeezing them. The cat was like feeling in danger of its life. And so it bit the child or scratched trying to get away. So usually it's from mishandling. The good thing about that, that means it's kind of rare. It's not something that we, worry about too much because most kids are supervised. They're not being that rough. So it's something we don't really worry about. We do try to keep an eye on it though with our veterinarian population. Because a lot of times when you're giving cats shots or treating them, they're not in the best of moods. So they can be, prone to getting bites or scratches as well. Now, with cat scratch fever, what usually happens is you'll start to feel a general malaise. The area on the skin will start to scratch a bit and will form a pustule. So it'll form like a little bubble or a pustule. And then what will happen is your lymph nodes will start to swell. Lymph nodes are areas where bacteria or viruses can be pushed to in the body to try to be destroyed by our defense systems. And so you'll see that the lymph nodes, usually behind the ears or at the neck region, will start to swell. Lymph nodes are really interesting. They give us a hint if we're fighting an infection. If you ever go to the doctor and say you're not feeling good, one of the things they're going to do is feel under your throat. Have you ever wondered, what are they doing under there? They're feeling for your lymph nodes. They want to see if your lymph nodes are swollen. If they're swollen, that can indicate that you're fighting bacterial or viral infections actively. So that's what they're checking under your throat for. Now, again, usually this is a problem that can be cleared up quickly. And the interesting thing about this organism with Bartonella is that it comes and goes with cats. It seems like some cats will be naturally colonized with it, and then as they age, they lose it. So that's why we don't do it as a regular screen. We'll find cats sometimes will have Bartonella in their saliva for like maybe a month and then it's gone. So again, we don't really worry about it because it tends to be almost like a normal flora with them. But as long as they're not feeling scared and doing deep bites, we really don't worry about it. So it's something that we just kind of keep an eye on. Let's go to our beta group. So those were our alphas. Let's talk about our betas. Betas are kind of like the pampered princesses of the bacteria world. These guys are really, really needy when it comes to their nutrients. And a lot of the organisms in here are major pathogens. A lot of the pathogens we'll talk about you'll recognize. So what I mean by needy, they need to have a lot of nutrients available to them. They need to have a lot of resources in their environment as they're growing. And so human cells or animal cells are wonderful for them because we provide everything for them. When we try to grow them in the lab, they tend to be picky. We can grow them, but we have to grow them on special media that we have to put special ingredients in. So it's a lot of work to get these guys to grow. Like I said, they're the pampered princesses. So if they're found in the environment, they again tend to be found in areas that are very nutrient rich. So we would find them, for example, in sewage regions or in a peat moss area or at a decomposition site like a swamp, something that has lots of nutrients always coming into them. They have to have a lot of nutrients. Some of our pathogens that we worry about the most are going to be ones that are going to be our Neisseria group. So Neisseria gonorrhea, which is an STI that we just can't seem to ever get to go away, is one that we worry about. And Neisseria meningitidis is another one that we worry about. And we'll talk about what those cause in just a minute. Again, I have on the board kind of a little graphic showing you the different organisms and how we would classify the beta groupings. So how we would separate or sort them. So let's start with our first beta. Our first beta that we're going to talk about is Bordetella pertussis. Bordetella pertussis is a little gram-negative organism. Of course, it has to be gram-negative to be in the proteobacteria. It's non-modal, and it is an aerobic bacillus. So what does aerobic mean it needs? It needs oxygen. So Bordetella pertussis is going to be something that would cause infections in the nares and in the lungs because it needs high levels of oxygen. This is the organism that is associated with the disease called whooping cough. And we often have whooping cough outbreaks in San Luis and Santa Barbara County. This is why if you ever have a child, they will tell you to make sure anybody who's around your child has been vaccinated for whooping cough. If you work in the medical community, you are going to be vaccinated for whooping cough because we want to make sure that we don't get our children exposed to it. If you go to school, you're vaccinated for whooping cough. So what is whooping cough? Well, basically, it's a little organism that can cause a lung infection in the cilia of our lungs. So cilia are little hair-like extensions that line our lungs. When we breathe in and we have debris or when we have any kind of a mucus that forms in our lungs, those cilia will grab it and they'll move it away from the deep lungs and cause us to cough to try to get rid of whatever that infectious agent is or whatever that dust or debris is that we breathe in. Unfortunately, with these guys, what they do is they get into our lungs and they paralyze the cilia. So they can no longer move or try to get debris out. So what that means then is we get this buildup of phlegm and mucus in the lungs, which is very difficult to control and to get rid of. So people who have whooping cough, adults, when they cough, they'll do this kind of like sound, kind of trying to clear out their lungs, but it's like they can't get a deep cough. It's really difficult because everything's just stuck in there. Adults are lucky though, because at least we have the muscle tone to cough, even when we, have a hard time doing it, we have the muscle tone to be able to do it. Where we worry about it the most is with our infants. With infants, we can't tell the difference a lot of times between a cold or whooping cough in the beginning. They're lethargic, they have a slight fever, so we're like, okay, it could be a cold. Unfortunately, what happens for them though is as that mucus begins to build up, they are unable to clear any of their lungs. They can't do a big cough. And so what will happen is that you will check on your baby and they will be turning blue and unable to breathe. And it can be life-threatening. So it can be something that will cause your baby to stop breathing. So we watch for apnea. Apnea is where suddenly the baby will just stop breathing on us. And it's because they have so much mucus inside of their lungs, they just can't clear it. And they're too small to cough. When a baby has whooping cough, they actually have this characteristic high-pitched whooping or wheezing sound, and that's where it gets its name from. They do this woo, woo, like they're trying to cough, but they just can't clear it. Adults don't do that whoop. It's like you can have whooping cough without the whoop. Okay? So we have like a deeper kind of cough as we're trying to clear it. Here's a picture of our little Bordetella on here, and that would be in orange, and these are the cilia that is attacking and paralyzing. So you can see the cilia are all being attacked by that Bordetella, and they're going to cause paralysis there. And so again, the cilia aren't going to be able to work. They're going to fill up with mucus, and it's going to be hard to clear them. We can do antibiotics, but unfortunately, with the antibiotics, a lot of times it's almost too late when you have a baby or an infant come in, because again, they can't tell you that they're not feeling good. And so that usually means a hospital stay. They're definitely going to be there for a while, and a lot of times it can result in life. I have a student who mentioned on here that they had a cousin whose infant died from whooping cough. So it's something that happens, unfortunately, fairly frequently. And so a lot of times, it's one of those things why we really push the vaccination for it because we can prevent it. The next group we're going to talk about are oh yes, go ahead.
How old do they have to be to get the vaccination?
So usually for babies, they have to be at least over a year old. And that's one of the problems is when they're in that infancy, they're the most exposed and the most vulnerable, but they can't get the vaccination yet. So usually they'll recommend doing vaccinations at around the year mark. Yeah.
And it doesn't require boosters later in life?
No, not usually. I shouldn't say that. As a child, no. It works for them through childhood, but as adults, yes, we do need to get ourselves boosted. Because one of the things that happens with vaccinations, I mentioned they're like most wanted posters. As we age, those posters fade And so our bodies tend to forget them. So that's why we have to get a booster to remind our immune system. So adults do, like when you go into school, I think it's in 6th grade, you get your booster again. Yeah. How do you get it? You get exposed from people who are coughing and spreading it. So when people finally are able to cough, mucus will come out, and that mucus will protect the organism from drying out. And then if you touch a surface, or if you're around that person and inhale that aerosol, you're going to get exposed. And that's one of the things we worry about with infants. If you're holding an infant and you're positive, you're going to easily transfer it to the baby. Big question. Let's talk a little bit about Neisseria. So Neisseria gonorrhea and Neisseria meningitis, these are going to be organisms that are going to be what we call fastidious. Fastidious means difficult to grow. So when we grow these in the laboratory, one of the things we find is that we have to give them perfect media. They like to grow on this media here. It's called chocolate agar, which actually sounds really good, right? I'm like, yeah, I grow on chocolate agar too. But the chocolate agar that we give bacteria is actually hemolyzed blood. So it's like a blood agar plate that has been heated or treated in some way to open up the blood cells. This is how pampered princess they are. I mean, they're so lazy, they won't even open the blood cells themselves. We have to spoon feed them to them. That's what we have to do. That's what chocolate agar is. So we have to open the blood cells and then give it to them. I mean, it's that kind of thing. So they are going to grow on chocolate agar. When they grow, if we do a gram stain on them, you can usually tell a Neisseria pretty quickly. Even though they're gram negative, like a whole bunch of other organisms, they always have this characteristic like double bean shape. They look like 2 kidney cells or 2 kidney beans, or they look like 2 coffee beans. They always have this little double bubble together. So when you see these little double cells, you know that you have a Neisseria that you've isolated. So whether it's gonorrhea or meningitis, they're going to have that double. So and it's a diplococci. We worry about Neisseria, as I mentioned before, because it's an STI. It can be spread through sexual intercourse. The reason we worry about Neisseria, though, a lot recently is because it is becoming very resistant to all of the different treatments that are on the market. And so we have an issue right now where a lot of strains that are circulating as an STI are antibiotic resistant, which means that once you get exposed and get it, it's hard for your body to clear, and we can't help your body with it. Now, why we worry about Neisseria gonorrhea as an STI, a lot of people, they won't have infection signs. They might have like a little discomfort when they pee initially, but then as the bacteria kind of settles in their body, some of their signs and symptoms go away. But we worry about it because what it does do is it causes repeated inflammation and scarring in the reproductive organs. And so if you get exposed as a teenager or as a young adult in your 20s, and then you decide maybe later in your late 20s or 30s you want to start a family, you're going to have issues becoming pregnant or starting an offspring because of that swelling that's been happening off and on that you weren't even aware of. This is like kind of behind the scenes. With that Neisseria infection, it can cause infertility. And so that's one of the reasons why they screen for this a lot. If you have like an annual check, they'll check if you're sexually active, they'll check to see if you have gonorrhea. The other thing that they will screen if you're pregnant is for gonorrhea. And the reason for that is because if you are pregnant and you're going to give birth, your child will pass through your birthing canal. And if they pass through your birthing canal and you have a gonorrhea infection, they can get exposed. The picture of the child's eyes crusted over is showing a gonorrheal infection of the eyes. So as the baby passed through the birth canal, it was exposed to gonorrhea. They have no immune defense. And so as they're born, the gonorrhea immediately starts attacking them. And it usually likes membrane surfaces. And since it can't get to their genitalia, it will attack their eyes the easiest. This, unfortunately, if untreated, can result in blindness. This is why we used to put silver nitrate drops in infants' eyes. The silver, as we now know, is antibacterial. And so as soon as a baby was born, they would get these silver drops, and that would kill any gonorrhea they were exposed to. Nowadays, we tend to not do that as much because we do a lot of screening during pregnancy to make sure that the mother is not positive for gonorrhea. But if we have somebody who comes in who maybe doesn't know they were pregnant, it's like a last minute thing, or they haven't had prenatal care, then we will do the silver drops because we want to make sure that that baby doesn't get exposed. Yes?
Can this be avoided through the cesarean section?
Yes, so if you have a C-section, one of the things that we have noticed is that you tend to not end up getting Neisseria. However, if you have an unsterile hospital environment, so if you think about where they're cutting here, if you have fluids exchanging, there is still a low risk. So even if you have cesarean, they will be careful. And a lot of hospitals, if it's a C-section, they will still do the silver nitrates just to make sure, because there's too much of a risk. Because we definitely don't want to have a baby being blinded for, exposure. Now, another organism that's in this group is the Neisseria meningitidis. When you hear meningitis, what do you think of? Meningitis. Yeah, meningitis. So this is an organism that can cause the spread of meningitis. Usually, again, it'll be spread through aerosols. So a lot of times these are associated with lung infections, and then when somebody coughs, it can be spread through an aerosol. If the person's immunocompromised or has a lower immune system, they can get the infection a little easier. It will cause swelling in the meninges, which are in the lower brainstem and upper spine, and it will result in stiff neck, high headache, high fevers, and can make life very uncomfortable. Again, they can be treated, but it will take time. And so far, our meningitis hasn't shown as much resistance as the gonorrhea to treatment. Gonorrhea, we're still kind of shaking our head at some of the strains that are out there. Let's talk about the gammas. The gamma group is going to be the biggest group of the proteobacteria. So a lot of our gram-negatives are put in this gamma group. And again, these are going to have a lot of organisms that can be pathogenic to plants as well as humans. So this tends to be our biggest group. So as you can see on here, we have some that are going to be autotrophic. What did that mean again? Self-feeding, good. And we're going to have a bunch that are chemoheterotrophs. And this is going to be the side that's going to be most associated with causing infections with humans. Our autotrophs, these are the ones that we mentioned before that are really good at helping with photosynthesis. So I don't know if you remember the first day of class, I mentioned we had a lot of bacteria that can help with photosynthesis. This side over here, our autotrophic group, that purple sulfur bacteria is one that's found in a lot of our waters that helps us with photosynthesis, that helps create oxygen for us. Over here, these guys are ones that a lot of times are associated with normal flora, and unfortunately, some of them are also going to be pathogenic. So let's start with the pseudomonids. The pseudomonids are going to be the ones that include the organism Pseudomonas. And you can see Pseudomonas growing at the top on a nutrient agar. Pseudomonas tends to have a really pretty greenish coloration when it's grown on certain types of agar. It just naturally will have that pigment to it. And Pseudomonas tends to be an organism that we do find a lot of times associated as being a normal flora. Now, the bad thing about Pseudomonas is it's something that we refer to as an opportunistic pathogen. What this means is that if it's in the right location in our body, like in our intestines, then it's harmless. But if it gets into a location where it shouldn't be, then it can cause an infection. So it's opportunistic. It will take advantage of an area. So for example, since it is found in our intestine, a lot of times it is found in our fecal matter. If it is able to move to the urethra, it will cause a UTI. So if it gets into the bladder that should be sterile, it will cause an infection. Same is true with E. coli. They're also opportunistic. So usually harmless, but in the wrong zone, we worry. Pseudomonas also tends to be an organism that's found in a lot of water areas. We find them, for example, in lakes, streams, sometimes at the beaches. And the reason we worry about that is because Pseudomonas, if it gets into the ear canal, can cause ear infections. This is why sometimes when you go to the beach or you go to a lake, they'll have a sign up that says high bacterial counts. Have you ever seen that? Or high bacterial loads. Don't know, swim at your own risk. If you do swim out there, make sure you don't have an open wound and make sure you don't submerge your head. Because if you do, you run the risk of getting bacteria in your eustachian canals and then you can get an ear infection, which is nasty to treat. Pseudomonas a lot of times is found in swimming pools. This is one of the ones that can cause infections when kids get things like swimmer's ear. They get water trapped against their ear and then the Pseudomonas that's in there will start to grow. So that's one of the things we worry about. A lot of these guys are also able to even grow inside of antiseptics. They're very resilient. We actually had an issue here on campus about a decade ago where our water fountain was smelling weird. It was like just this weird smell. And you guys might remember these from lab. Do you remember when I had you smell the Pseudomonas? Yeah, what did it smell like? Yeah, it just stinks. No, it smells good. It smells like tortilla chips. Remember Pseudomonas has a tortilla chip smell? Or tortillas? So Pseudomonas has this kind of a sweet smell. That water fountain, we noticed, started having a sweet smell to it. And so I swabbed it, and sure enough, it was full of Pseudomonas. So we tried to clean it, and it kept coming back. Now, the Pseudomonas was harmless. It would go into our stomach acid and die, so we were okay. But we needed to get rid of it. What we found was that it had created a biofilm. Remember we talked about biofilms before? It created a biofilm in the piping coming into the fountain. And so we could never really clear it because it just kept coming through our water lines. So we actually had to take the fountain apart, clear out all of the lines, and re-plumb and re-put them into place with antibacterial components. So it was kind of an interesting science experiment in action. So nowadays we do sample the water fountains all the time and they're safe. So that's a good sign. Yeah.
Does this bacteria grows at room temperature water, like tropical or?
Oh yeah, it'll grow. It's very wide range. So this particular organism, it'll grow at room temp, like 20 to 25, and this one will actually even grow up in our bodies at the 37. It doesn't like to go much higher than that. If I start pushing into the 40s, it's like, I don't like this anymore. But if I can grow up to the upper ends at 39 zone, it's fine. So it's one of those mesophiles that has a wide range. Remember we talked about how sometimes we have the overlap? This guy definitely has the overlap. Another organism that we worry about is an organism named Moraxella. Moraxella is known to cause conjunctivitis, which you might have heard of referred to as pink eye. So pink eye can be caused by either a virus or a bacterium. So usually when it's caused by a bacterium, it's usually Moraxella. And this one, unfortunately, is just really hard to clear. This is one that if you buy over-the-counter remedies, they're not going to work. And it's where your pink eye just keeps going on. It feels like it's never going to end. You have this itchy, irritated eye that's constantly bugging you. And eventually you have to go to the doctor to get a higher dose of antibiotic. So it doesn't work with OTC, over-the-counter treatments. It's just an inflammation of the eye lining, the conjunctiva. And so it's just going to cause irritation and itch with that eye. It can be spread from people rubbing their eye and then touching other surfaces or touching each other. So it's something that can be spread. That's one of the things that whenever we're using microscopes in here, I always am kind of looking around, seeing if everybody's eyes look okay, if they look red or irritated, because I have had that happen before. I had a student one time who I noticed was kind of rubbing their eye in lecture, and so I talked to her later during the break, and she was like, yeah, I think I'm getting pink eye, and I'm like, yeah, we're not going to do microscopes today. I'm just going to have you skip, we'll look at pictures later, because being in a microscope and touching the lens with your eyelashes could spread potentially the pink eye to other people. So we always have to watch that. Here's a picture of what our little Moraxella or Pseudomonas would look like. So again, they tend to be these little, they're shaped kind of like E. coli. They tend to have these bacillus shapes. They're gram-negatives and they have their flagella so that they're motile and can swim through the environment. This is what makes trying to tell what these organisms are so tricky. Pseudomonas, Moraxella, E. coli, they all look the same. So this is why we have to do those biochem tests that we were doing in here, trying to figure out what they eat and how they survive. Another one that we have random outbreaks with in the gamma group are the Legionella group. And the Legionellas are really interesting organisms that produce a really nasty toxin as they're growing. This is them growing on some black charcoal media. They're so toxic that they make themselves sick when we grow them on a petri dish. Their waste product and the toxin that they produce will actually cause their own death. So we have to grow them in this special media that has this dark appearance that is full of charcoal to filter out that toxin. And that's pretty bad. You have to be filtered so that you don't kill yourself, right? You have to have this charcoal present. Now, the Legionella group, they tend to like really warm water and kind of steamy locations. Where we first found them was during a Legionnaires meeting. And this Legionnaires meeting had gathered in Philadelphia. And the hotel that they were staying at for their convention, unfortunately, had a heated water tank that ended up getting infected with Legionella. We don't know how it got into the water tank. But when these people were taking their showers every morning and breathing in that steam and relaxing in that hot water, they were getting just a mass dose of Legionella on themselves and in the shower stalls. And so what happened was they ended up inhaling Legionella, which caused a lung infection in their body. So we all of a sudden had this mass outbreak where everybody who attended the meeting started showing signs of respiratory distress. It did result in a few deaths for people that were immunocompromised. Nowadays, where we see Legionella outbreaks is usually associated with things like hot tubs or saunas if they're not properly cleaned, or hotels, again, if they don't have their showers properly cleaned. This is something that now is looked for by inspectors. So you don't have to panic when you travel. Usually your inspectors that have been checking the hotels have been checking to make sure that everything is safe there as well. But before the Legionnaires Conference, we didn't know this organism was such a risk, and so it wasn't being checked for. There was an outbreak about 2 years ago at one of the local state fairs because they had a hot tub company that was going around, setting up hot tubs, kind of like when we go to Mid-State, they have hot tubs everywhere. I never really get the whole hot tub thing with the fairs. I don't know. It's just, when I go to a fair, I don't think I'm going to buy a hot tub. That's just me. But, some people apparently, they go to the fair to buy hot tubs. I'm like, okay, that's great. But so they had a hot tub situation set up at one of the fairs, and it ended up getting exposed to Legionella. And people that were leaning over, looking at the hot tubs, checking them out, ended up getting exposed, and we all of a sudden had a mass outbreak. So we see that a lot of times. So yeah, if you're at a fair and they have hot tubs set up, just you can stay a distance, go over and have a look at it, because that's harder to catch because it's not like you have as many inspectors there. Yeah.
This is under sauna. What would happen to you?
What's that?
This is under sauna. What would happen to your body? You said?
A sauna, so same thing. If you get exposed to them in a sauna, if you're breathing them in, then you run the risk of getting a respiratory infection. So same thing, you would get sick from them. It would cause a lung infection. But you gotta think, again, if I'm at an athletic group that would have a sauna, or a hotel that would have a sauna or shower, that I'm getting checked by inspectors, and so that's less risky. When they're on the road, it's harder. Does that make more sense? I don't have the same inspectors coming and checking things. Yeah.
When they're looking for it, what are they looking for?
They're going to do growth. So they're going to do a swab sample, a water sample, and see if anything grows on their media. And this media is going to be used as selective, so only the Legionella can grow. And it has that charcoal component, so we can actually get germination.
What is that?
This is Legionella.
So the media.
This media is called BCYE. It's a charcoal component media.
Okay.
Another organism that we have out there is one that's referred to as Q fever. This is one that, again, is transmitted through aerosols. It's one that tends to be fairly rare, but the reason I bring it up is because it's one that's associated with mass fatigue. You feel like you're run down all the time. I feel like I always have this. I think I have Q fever. I'm so tired. But you feel like you're run down, but you also have a fever, a cough, just general malaise. This is one that a lot of times doctors aren't checking for. So if you ever have somebody who, is really feeling tired, they've been sick for a couple of weeks, they keep going in and getting checked and nothing is coming back positive. This is something then that you would ask, well, has your doctor checked for the organism that's going to cause Q fever, which is Coxiella? Again, it's fairly rare. We see it more over in the valley, the Central Valley area. But it's just one that's good to know in case you ever have a loved one that's having problems getting diagnosed with some illness that they have. So that's why I bring it up, is because we did have an instance one time where we had a student whose loved one was getting exposed and had unfortunately been exposed to Q fever, and they had it, and the doctor hadn't checked for it. And so then when they went back and asked to be checked, they were positive. So it's always good to know what's out there. Vibrio. So Vibrio has kind of been the bane of our existence since the beginning of time. Vibrio is a really old bacterium, and it's been around with us for so long. We have paintings and depictions of Vibrio infections from as far back as we have artwork. And Vibrio is an organism that is a facultative halophile. So what did that mean again? It can live not oxygen, but salt. Excellent, salt. Yeah, so it's facultative halophile. So it can either live in a salty environment or live in a freshwater environment. And that's what we find with Vibrio. They are found a lot of times in oceans, they're found in streams, they're found in lakes. Vibrio has this characteristic kind of boomerang appearance to them, or like a macaroni and cheese kind of a look to it, like a little macaroni and cheese noodle. And this particular organism is one that can cause really extreme gastroenteritis. What that means is stomach upset. So it's going to cause intense nausea and intense diarrhea. And we get exposed to it from drinking contaminated water. So the water would have Vibrio, we drink it, and this organism is not affected, unfortunately, by our stomach acids. So it is able to move through to our intestines, and it causes a nasty infection. Now, it's a facultative anaerobe. That means it can live with or without oxygen. So it can live inside of us or outside of us. And usually the process is that we will have an exposure to Vibrio from the consumption of contaminated water. So I have somebody who gets sick. That person starts to show signs of Vibrio by having intense diarrhea and upset stomach. Here's a drawing from a really interesting book. If you ever want to read an old medical literature book, it's called The Sick Rose. And it's a book that's about 3 years old that basically went around and gathered medical drawings from like the very beginning of medicine and compiled them all into this book. So when you used to study medicine, textbooks and patients sometimes were hard to come by. And so a lot of times when you were studying medicine, they would show you literature, they would show you drawings and different pictures of diseases. So this book is kind of like a walk down horror lane. It shows you everything that could go wrong with the body, like every type of deformation that's out there. But it's a really fascinating book because it's all these medical depictions from 1600s and on that have been preserved. This is one that's referred to as the Vibrio depiction. So this one was done in the 1800s. A young girl that came in was showing signs of stomach upset. So they did a quick sketch of her. They said, she looks healthy. She is having diarrhea, upset stomach. Other than that, nothing else. This was her 2 days later. So skeletal drawn in, a pyosin or blue coloration to the lips or skin. And what's happening here is that her body is super, super dehydrated. It's starting to sink in on itself, so we're now able to see all the venation. We have a loss of pigment to the skin because we're so exhausted and so dehydrated, and so we look really pale and wan. Now, the reason we worry about Vibrio then, and this is an old depiction of Vibrio coming in and harassing Roman soldiers. This is how far back it goes. It goes way back to gladiator times. It goes back to the Greeks. It goes way back. And so here with Vibrio then, there's really not a treatment of antibiotics that we do. Instead, we do a treatment of fluids. So the job when somebody gets exposed to Vibrio is to keep them hydrated. So here's a little girl who came into one of the clinics in Haiti. You can see she's got this sunken in appearance. She's got, it's kind of a skeletal look to her. And that's because she's just so dehydrated. And so the typical treatment is to do a saline IV pack as fast as possible. And then just to basically try to keep them hydrated while this Vibrio clears their body. There's not really anything else we can do. Antibiotics, they're not really going to be able to help. This is her just a few days later. Isn't she gorgeous? I love that little smile. So that's her just a few days later, full recovery after getting IV. So what does Vibrio do? Well, it gets into our intestines and it attaches to our intestines. So it sticks to our intestine. And our immune system basically goes into a full-on panic. It's kind of like, have you ever walked through a spider web? Yeah, a few of you have. Have you ever done the spider web dance? Where you're in the full-on panic. You're like, oh, and you're like doing that. That's what your immune system does. It's like, oh, there's Vibrio here. And so the Vibrio itself, it can produce a small toxin, but it's not enough to really cause our immune systems to react as much as they do. But it's like an overreaction. Our immune system goes into full panic. So it's like, okay, there's something in the intestine, hit the eject button, and it's out of here. So what your body starts to do is start having massive diarrhea to try to get this thing out of your intestines. The problem is Vibrio is very sticky. So it's sticking to the intestinal walls. So you have diarrhea, you have it again, you have it again. You get to the point where your diarrhea looks like at the top. It is colorless because you're not even having time to produce bile. Bile is usually what gives diarrhea its color. It is also referred to as rice water stool. And the rice water stool, if you can see, there's like a few little white pieces kind of floating in there. I know it's gross to look at diarrhea, but in this picture, the few little white balls that are floating in there, those are pieces of your intestine. Your diarrhea is so strong, it is sloughing off the inside of your intestinal lining. That's like the full-on, it's the spider web, full-on panic thing that we got going on here. And so we call it rice water stool because it's clear and it looks like pieces of rice in it. And so that's why we really can't treat what's going on in the body. There's not an antibiotic to give you because this will naturally clear within about 48 to 72 hours. If I start you on an antibiotic regime, usually you see me after 24 hours. By the time you get an antibiotic high enough, you're already cleared it. It's something that's already going to clear. So my job is to just keep you hydrated. That's all I can do, just to replace all those fluids that are going in there. Yeah.
Is the stools completely like water-like?
Yes.
Okay.
Yes, it's like totally liquid. There's no solid form in it except for those pieces of your intestine. And it's colorless. It's like chalky sometimes. It's like the weirdest looking thing you've ever seen.
It's acidic.
It tends to be acidic, yes. And a lot of times the beds in a Vibrio unit, they'll actually have you like on a cot and they just cut the bottom out of the bed and they just don't have you wearing anything and they have a bucket under. Because they can't even get you to the bathroom. It's just constant diarrhea. I mean, just pretty intense. So as you can imagine, when Vibrio would come to town, as this picture shows, it could be quite decimating when it would show up in a water supply. The way we usually get there is if some of that rice water stool were to get and spilled into a water source, it's full of millions of bacteria. If somebody else comes and drinks the water, they can't see it. It's clear. They can't tell what it is. And they're just ingesting this bacteria over and over. We are very fortunate where we live because our water is heavily regulated and heavily filtered. If you go to other countries, a lot of times they don't have the benefit of healthy sewage lines. They don't have the benefit of having clean water like we do. We take it for granted. And so in some countries, they have it where they have sewage lines that can be broken easily. So like in Haiti, anytime there's an earthquake or a hurricane or something along those lines, any natural disaster, we always see outbreaks of Vibrio. So one of the first things we do when we send relief is we send a ton of saline packs. So that way we can try to keep people hydrated as much as possible. Let's talk a little bit about Pasteurella. So Pasteurella is another organism that's in this gamma group. We'll talk more about it on slide 29, so coming up. It basically is known to cause a pneumonia. And the reason we worry about Pasteurella is it's an organism that can easily go septic on us very quickly. So we talked about septic, having sepsis in here or having septicemia. Remember how we talked about how that could be deadly? It can have blood clots. It can be really decimated to the family or to the body. This is an organism, for whatever reason, that will cause disease and then quickly spread in the bloodstream. We're still trying to figure out what it is that makes it so readily able to move to another location. We also in this group have an organism referred to as Haemophila. And Haemophila is a really interesting organism. It is required to grow inside of a host for something that is its component for iron. We call that an X factor, and for a V factor. What these are is they're precursors for NAD and NADP. Where do we use NAD and NADP? You remember? Where do we use NAD? Yeah, in our respiration process, right? Remember we had glycolysis, Krebs, ETC. We also had NADP that was running along the sides of the pentose phosphate shunt was one of them that uses it. So these again are Uber drivers. This organism, Haemophila, is not able to make its own factors or precursors for NAD or NADP, so it always has to be in a host cell. If I want to grow it on a petri dish, I have to give it these factors. And so what we usually do is we put little discs on the petri dish when we grow the bacteria. They have what are called X and V factors. They diffuse in the media to give the organism what it needs to be able to grow its NAD. It's pretty fascinating that we have these little things that we have to do to keep these organisms alive. In the gamma group, we also have what are referred to as the Enterobacteriaceae. We mentioned these in our labs when we talked about having the use of our McConkey media as well as our EMB media. There was a mention of the Enterobacteriaceae group. These are also referred to as the enterics. And these are the organisms that are found a lot of times in our intestines. A lot of these can be associated with normal flora. A lot of these can be associated with illnesses. So some of our normal flora that's up here are things like E. coli, Proteus is another normal flora, Klebsiella is another normal flora, Enterobacter is another normal flora. Those are all ones that are normal. Some of the pathogens that are up here would be things like Shigella, Salmonella, Serratia, and Yersinia. Those would be ones that we wouldn't want to see, but that can easily grow if they get exposed in our intestines. They all are going to be facultative anaerobes, and they all have paratrichous flagella. What did that mean again? Yeah, bad hair day. They have it like everywhere, right? So they can really move. So here's a picture of the flagella. It's everywhere. One of the ones that we saw in here on our first day of class was Proteus. Some of you have Proteus when we did our handprints. And the Proteus is one that can be found a lot of times on surfaces. It's just easily found. It's the organism that tended to spread out a lot or to swarm out. So Proteus is another one that as it grows, it makes itself sick. It produces a little toxin that makes itself sick. And so what it'll do is it'll make a colony, and then as the toxin or waste products build, it'll kind of spread out from itself and then keep spreading out and keep spreading out. So when I grow Proteus, a lot of times they'll just cover the entire plate like a little lawn. They just grow all over the plate because they're trying to run away from themselves. Again, how bad do you have to be to make yourself sick? You know, I mean, that's pretty intense. So one of the ones that we can talk about here is Salmonella. We have two different varieties of Salmonella, or actually we have a lot more than that, but two main ones. Salmonella enterica is one that is associated with salmonellosis. Also, we have another organism, Salmonella, which should be in this group. These are ones that a lot of times are associated with fecal matter coming into contact with food. So the reason we worry about eggs, we talked about removing the bloom and then having Salmonella on the surface of eggs. They can then get in through the porous eggshell. Also, if it's on the surface of the egg and if I break it and it falls into my cake batter, then I can end up, infecting myself that way. Another way I can get it is through fecal exposure if I have dirty water that I'm drinking. That would be my Salmonella enterica. The other one I worry about is Salmonella typhi. So Salmonella typhi was associated with typhoid fever. It would cause a high fever, general malaise, respiratory infection, and then usually if it would get high enough and left untreated, it can result in death. So Salmonella typhi was something that a lot of people lived in fear of in the early 1900s when there wasn't a treatment for it. If you got exposed to Salmonella typhi, you had a very good chance of dying of typhoid fever. Now, there's a really interesting story you might have heard about from a woman named Mary Malone, who is referred to as Typhoid Mary. Has anybody heard of Typhoid Mary? What is Typhoid Mary famous for? She's famous for typhoid. I love that. But how? Does anybody know her story? So Typhoid Mary was known as being a plague spreader, which is really sad. It kind of is a sad story. So Typhoid Mary was a woman who ended up becoming contaminated with Salmonella typhi, but she showed no signs or symptoms. So she was what we call a carrier, which means that she could spread and pass it, but she had no signs or symptoms herself. She was perfectly healthy. So what happened with Typhoid Mary is that she was hired as a cook. And during the late 1800s, 1900s, to be a cook was a huge thing in a house. That was like a very prestigious position. And she worked really hard to get that position. Unfortunately for her, whenever she would prepare food that wasn't cooked, so think about like cutting fruits or cantaloupes or making ice cream or making things that are like desserts that aren't being cooked, she ran the risk of passing on Salmonella typhi. And she was known to wash her hands. She was known to be a clean person. But the problem is when you have rings or when you have anything on your hands, it allows bacteria to hide. So even if she was scrubbing, she couldn't get rid of all of the bacteria that would stay with her. So her story starts where she ends up cooking for a very prestigious family. Their children end up getting exposed to Salmonella typhi. They get typhoid fever. She's taking care of them. She's cooking for the rest of the family. And eventually the entire family of 6 died from typhoid fever. She never showed signs or symptoms, so she was released. And when she was released after the family died, a family down the street who had had her cooking and loved her hired her immediately. And then guess what happened to them? Yeah, they ended up dying. So that family died. So now the police came in and they're like, wait a minute, you're at this house that dies, now you're at this house that dies. Are you killing them? They do an investigation. Can't find anything. They let her go. She goes to a third house and guess what happens? They die too. So now we have 3 families dying of typhoid fever. So they're trying to figure out what's going on. While she's at the police station, they do a fecal check and they find out she's positive for Salmonella typhi. This is the first time we actually had evidence of a carrier. That was the first time we found somebody who hadn't really shown signs or symptoms but had the disease. Now, Mary didn't agree with this. She paid to have her own testing done. And when her test came back, when she paid for it, it was negative. So what was the deal here? Well, it turns out Salmonella typhi naturally has this issue where it ebbs and flows in the body with their defense system. So there would be times when her Salmonella would be high, and then there were times when it was low. So the police caught it at a high, and she caught it at a low. This is also why she could cook for families for months and months and not show any illness, and then all of a sudden they would get sick. It was because she was ebbing and flowing. Her story goes on, unfortunately, to the point where she was told she could no longer be a cook. She was told she could be doing something else. But again, a cook was a prestigious position. So she decided to secretly change her name and become a cook again. So she did. She changed her name. She became a cook again because she didn't believe the results. She had a low test. She started cooking for a family, and guess what happened? They tragically died as well. This time when the police showed up, they said, "We're done. We obviously can't trust you." So what they did is they shipped her to an island off of New York, and she was isolated and forgotten about there. However, the medical community kept her there and were running tests on her constantly to see if they could get rid of the Salmonella. She was given arsenic. She was given mercury. She was given every kind of treatment under the sun that they could think of, trying to figure out a way to cure her. And she ended up dying forgotten about, alone on this island. It's just a scary story because it's something where, a lot of people will say, oh, she intentionally got people sick, but you can understand her reasoning when you learn about her story. And just the fact that she was shipped somewhere and then experimented on is just like a horrific story. I mean, this should be like a Netflix movie, don't you think? Shouldn't this be like a movie? Yeah, you had your hand up.
I was just curious, that's kind of weird, why everybody died and she's
Again, she just thought it was like they were getting exposed somewhere else because when she did her own testing, it was negative. That was the thing. It's just like, she was like, well, I'm not the cause. Like, my tests are negative. They're obviously lying, So it was just a big mistrust on both sides, unfortunately, which resulted in a lot of death. So this is an expression now. When somebody is sick and spreading disease, you'll sometimes hear somebody say, oh, they're a typhoid Mary. That means that they're a disease spreader. That used to be a common expression that was used. Let's talk about a few more enterics. So, as I mentioned before, we can have E. coli, Escherichia. We can also have our Enterobacter group. We've worked with Enterobacter aerogenes in here. Those are ones that are normal flora that are in our bodies, but again, they're opportunistic. If they get into the wrong place, they can cause an infection. So if they get in the bladder, they can cause a UTI. I love it. Yeah, Michelle says she agrees. She can see she have a Netflix series on it. Serratia is another one that is, again, found a lot of times in the body. That was our pretty orange pigment that we grew in the lab. Remember our pretty red or orange pigment? Was our Serratia marcescens. That was on your lab quiz. This is an organism that's really interesting. That red pigment that it created in the lab, it will only create that if it's at a temperature under 30. If I was to crank up and put it in the incubator at 35, it would actually lose that pigment. That pigment is temperature sensitive, which is kind of interesting to see. I had a friend of mine who one time was teaching a micro lab, and they were trying to grow Serratia, and she called me in a panic because their Serratia was white. And she's like, I don't know what it is. It's not Serratia. I'm like, well, what temperature did you grow it at? And she's like, 37. I'm like, well, yeah, you knocked the pigment out. You got to make it colder to get that pigment produced. Shigella, that's another organism that we worry about because that can cause something referred to as dysentery. So again, that means intestinal upset. So it can cause dysentery. Proteus, we talked about our swarming colonies of Proteus and how it can cause UTI. The one here, Yersinia. Yersinia pestis, this is the organism that was associated with the spread of the Black Plague, or the Black Death, that decimated most of Europe. The Black Plague we now know was spread by what?
Rats.
Not by rats, but what was on the rats? The fleas. Yeah, so it turns out that the Black Plague has a really interesting story. The Yersinia pestis, the organism that causes Black Plague, it will get ingested by a flea, and it will block their feeding pipe. It will block their food pipe. So the flea is slowly starving to death. And so the flea will keep going and bite other hosts, cats, dogs, humans, whatever it can bite, to take a blood meal. When it bites that host, it vomits. It does like a throw up. And the Yersinia that's in its throat will come out and infect the person that it bit, or infect the dog or the cat that it bit, or the rat. And then that flea is still slowly dying of starvation. It keeps biting more and more frantically, and so we keep spreading Yersinia more and more and more. Now, rats were natural carriers because they were usually covered in fleas. When Yersinia pestis was around in communities that had the plague, one of the things that they did is they didn't know how it was being spread. So immediately they blamed cats and dogs. So they slaughtered their cats and dogs, which means that now the fleas didn't have as many hosts to choose from. So the fleas started going on humans. It actually made the problem worse. The other thing was that we ran into is that when people would have the Black Plague, they would tend to have these swellings. They're called buboes, and I'll show you pictures of them later. It's where your lymph nodes swell with the bacteria. But because this bacteria was such a nasty one, it would cause cell death. So your lymph nodes would swell, and then they would start to turn black. That's why it ended up being called the Black Death. So you get these black colors to your skin. One of the treatments that they tried doing was popping those buboes, those lymph nodes that were swelling. And if you pop them, what you do is you release a huge amount of bacteria in the bloodstream, which is causing sepsis. So it ended up resulting in a lot of death. We found out that the plague is also the first instance of warfare between countries, bioterrorism, where they would take plague victims and they would catapult them over the walls of the towns they didn't like. Can you imagine this? This is pretty horrific. You have somebody not feeling good. You're like, oh, I know you don't feel good. Here, sit down right here. Okay, we're gonna line you down. Just stay right here. Okay, just stay right there. Don't move. Okay, boing, and then they'll launch you over a fence. I mean, this is horrific times, right? And as soon as that person landed, of course, people would come running out trying to get rid of them, trying to move the body, and they're unfortunately releasing the plague. So it's kind of a scary thought. We still have a song that is sung today that we think is in reference to the plague. Anybody know what it is? Ring Around the Rosie. Yeah, so Ring Around the Rosies. How does it go? Ring Around the Rosies? Well, that would be the ring around the lymph nodes that would form. A pocket full of posies. Remember we talked about miasma? What do we carry in our pockets to keep bacteria away? Flowers. Pockets full of posies. Ashes to ashes. That means that we would be burning the victims, right? We wanted to make sure we got rid of the plague. If somebody died, we burn the bodies. And we all fall down means the entire villages were getting wiped out. Isn't this lovely? We teach this to our children, right? We're like, okay, let's sing the plague song. It's like, but it's a fun song to sing, but it's kind of like, it's interesting the origin or where it comes from. Now, of course, there are some that argue and say, well, that's not true. It's not. But for the most part, we're finding that it showed up right when the Black Plague came around.
Yeah, that's easy to hygiene too.
So the hygiene, it was always kind of there, but for Yersinia pestis, it didn't really matter. It was more fleas trying to take a blood meal. So even if you were clean, fleas would jump on you. What's funny is, do you guys remember we talked about the masks that the plague doctors wore? They were full of flowers and stuff. There's a really interesting notebook that was recovered about a plague doctor talking about his experiences during the Black Plague, and he survived. He talked about wearing that mask and how hot it was and his long leather cap and gown thing that he wore. And he said these things are uncomfortable, but they're mandatory. We need them to treat our patients. And he even says in there, the only thing they really seem good at is that they keep the fleas off of you. That doctor said what was the causative agent and didn't even realize it. He survived, and he said the only thing the leather did was keep the fleas off of you when you would go into towns or villages. That's what kept him alive. Isn't that funny? He had the key. He knew what it was, but he just didn't put it together. So that's why, when we take notebooks or when we do work in science, we always tell people write everything down, because sometimes you'll write something down that'll be like a key later. And you're kind of like, oh, that's interesting stuff. Okay, let's go on to the next group, are still in our gammas, but these are the epsilon gammas. So these are going to be like our pastorellas. Pasteurellas are known to cause pneumonia. I told you that was coming up again on that slide, so this is what we're going to talk about Pasteurella. They cause pneumonia. And I mentioned in the previous slide, the reason we worry about them is because they also tend to go septic very quickly. Usually Pasteurella is associated with dog bites or cat bites. When we have somebody who's Pasteurella positive, we're going to treat them right away. So when we have somebody come in with a dog bite or a cat bite or even a human bite, one of the first things we check for is Pasteurella, because it could start causing a lot of tissue damage and breakdown at the site of the wound. We also worry because they can go septic so quickly. So we have kind of like a few, maybe like a day or two at the most to try to get this thing under control. We mentioned Haemophilus before, how they love heme. Those are ones that we have to grow with those X and B factors. We mentioned that these are the ones that have to have different factors to help them grow so they can create their NADs and their NADPs. Their name, Haemophila, comes from the term loving heme or loving blood, because it turns out that that's where they get their iron that they need to make their NAD and their NADP. Here's another one that we're seeing occasionally popping up over in the Central Valley. And this is Francisella tularensis. And this is known for causing tularemia, or sometimes it's called rabbit fever, because we would see it in wild rabbit populations. And what it is it's where a rabbit is usually the natural carrier for this illness. They just pass it back and forth to each other. But sometimes you might have a flea or a tick that's fed on that rabbit, get into contact with a human, and then feed on you and spread the tularensia. And what will happen is it gets into our bloodstream and it will cause a pneumonia-like infection. It tends to be one that also causes a very high fever. So high fever, pneumonia, general malaise. Again, it tends to be one that is fairly rare, but we're starting to see a slow increase in the Central Valley region. Here, our numbers are still really low. If we find a positive here, it's usually from somebody visiting from the Central Valley region. So what we're thinking is as we're spreading out and building more homes, we're kind of taking away some of the natural habitat of our wild rabbits, and we're getting more exposed to the diseases that they carry, which is making this more of an issue as we continue our urban spread or urban crawl.
Are there vaccines for the rabbits?
No, not yet. This particular organism, it's been pretty dicey to try to make a vaccine for. We're trying. But it's one of those things too where there's just so many rabbits, it would be hard to capture and vaccinate them all. We don't see it with domestic rabbits. Okay. So if you have a rabbit that you've adopted or you're born and bred in captivity, it hasn't been out in the wild, we don't see it.
Would they show like signs and symptoms?
Yes. They would show respiratory infection, they'd be lethargic, they wouldn't be eating. I mean, you would know they were sick. Yes, they would definitely be ill. If you have a pet bunny at home, don't worry about it because they're not out in the wild. It tends to be something we see circulating in the wild population.
So just keep an eye on like fleas and ticks and stuff like that.
Yeah, exactly. And again, even the fleas and ticks around here, they don't carry it. We tend to see it more in the Central Valley regions. So that's where we worry about it more. So if you have your rabbit and he's outdoors, don't worry about him. Like he's not going to pick it up here. Yeah, he wouldn't pick it up here. So it'd be pretty rare. It's kind of like the plague. I didn't mention this, but the plague is still spreading in wild animal populations. So, for example, ground squirrels can carry the plague. That's why family members always tell you don't pet ground squirrels. It's because they naturally carry the plague, and prairie dogs.
Yeah, I read and you said that.
Yes, Yosemite has one of the highest plague rate issues. And again, it's not affecting humans; it's affecting the wildlife. They're spreading it back and forth. If humans aren't getting close to the animals, then we don't have to get close to their fleas. We don't have to worry about getting exposed to the plague. So as long as we keep our distance, we're fine. But it tends to be in their populations because they're burrowing together, they're close together, so it tends to be more in wild populations. The last time we had a bubonic plague outbreak, we actually had two. One was in San Francisco. It was because a person ran over a ground squirrel with their lawnmower. Like, why you would do this? So the ground squirrel was dead. It was on their lawn, and rather than stop and pick it up and throw it away, they were mowing the lawn, and they were like, "Hey, I'll just run over it." So they ran over it. And so, of course, blood splattered, and they inhaled some of the Yersinia pestis that was in the ground squirrel. It's like, don't do that. Don't run over an animal. Yeah.
Rabbits or regular rabbits with this defect.
Yeah, so we're seeing it primarily in just the regular rabbits, not so much the jackrabbits. We see it primarily in just the gray rabbits and the natural rabbits that are out there. You're right.
I forgot if you mentioned in a previous lecture whether most people have antibodies against Yersinia pestis.
Excellent. So you're absolutely right. So there's different strains of Yersinia pestis out there. There's one that's a pneumonia strain, and then one that tends to be more of a skin and blood infection strain. The pneumonia one is the one that was decimating Europe, and that particular strain, a lot of us have natural antibodies too. So excellent point. But it's also where we have to be careful because there are other strains out there, kind of like COVID, where you could still get exposed to another strain.
Is there a treatment now?
There is a treatment, so you can get treated. It just depends on which one it is. If it's the pneumonia-based one, you can get treated, but it's like COVID, where you can end up going into the hospital and have to be on a ventilator because it can cause mass lung tissue damage. The reason for that is some people's immune systems, again, kind of go into the spiderweb thing. They go a little overboard, and that's what we see with COVID, too. Same idea. Some people's immune systems go a little wonky. We'll talk about that when we get to our viruses. So the other outbreak that we had with plague was in LA, and it was from a ship. It was a company that was shipping in some of the big crates that we see down there at the harbor. And unfortunately, the ship, when it arrived, had a bunch of crew members that were sick. They were sick with the plague. They had rats on their ship. They had the plague. And then when they landed, they went to the hospital. They had talked to all the workers, the dock workers. The dock workers went on their boat to help them since they weren't feeling good, and they got exposed to fleas, and then they ended up getting the plague. So that was one of the other outbreaks. All right, any other questions on that? You're asking me a question. Okay. Did I talk over? I have, huh? I should stop. Yeah. Sorry, I'm just like excited to hear this now. I'll just keep talking forever. Okay, let me stop. We'll stop talking now. And then let's see. Since we usually start out at 10:30, let's start out at 10:35. So that way you get your full 10 minutes. Okay.